The second part of the lecture on vision includes consideration of light perception in the retina and the visual cortex. Retina of the eye can be seen using a special device called ophthalmoscope. There is a typical view of retina, and its schematic presentation is given at the right side. The main structures visible in the retina are the optic disc, which is this area where the optic nerve leaves the eyeball, and also artery and vein for the eye pass here. The other structures are macula lutea and fovea. Macula lutea is a circular region in the retina about 3 mm in diameter that has high visual acuity due to high density of photoreceptors. Macula lutea is translated exactly as yellow spot. The yellow color is due to high content of yellow carotenoids, lutein and zeaxanthin. These substances, due to their yellow color, enable the macula to absorb excess of blue and ultraviolet light, which makes it something like the natural sunscreen. The fovea is a central area, about just half of millimeter in diameter, and this is the region where density of photoreceptors is highest. Actually, there are only cones located here, and they're packed very densely, so many thousands of cones are located in this little area. Actually, both macula and fovea consist of many concentric subregions with some functional differences, and that's why you can find some discrepancies in the description of the sizes of these structures. And mostly fovea, under fovea, the central fovea region is understood. Clearly, it's exactly half of millimeter. And exactly in this area there is a pit, an indentation in the retina, and fovea exactly means a pit. Let's consider the main cells of the retina. Here you can see the photoreceptors. They are rods and cones, and this picture shows only rods. Then, next cells the rods are connected to are bipolar cells, and it's easy to memorize that they are located between, because bipolar, one pole is directed to rods and the other pole to the next cells, ganglion cells. And only the last type of cell, ganglion cell, produce action potential or nervous impulses, and their axons form the optic nerve. Also here you can notice 1, 2, 3 layers, and here you can notice horizontal cells inhibitory. In many cases, horizontal cells provide contact between rods and bipolar cells. Not always, there only 3 cells are included into the full levels of conduction. So sometimes between rods there is a horizontal cell, inhibitory cell, and between rods and bipolar cells. Also there are some other inhibitory cells, amacrine cells, but we'll consider later. Let's recall layers of retina. To the right side you can see the names of the layers considered in histology, but we are not much interested in these all numbers and names of the layers. And again, let's observe the main cells. Photoreceptors, rods and cones. Cones are really thicker and have conical shape. You can see the photosensitive parts of these cells are directed for this picture upward. Then they contact to bipolar cells and finally to the ganglion cells. And you see optic nerve is formed by ganglion cells. Then there are horizontal cells that provide contact between some photoreceptors and bipolar cells, and in lower level, in a plexiform layer actually, amacrine cells are located, also inhibitory. And let's recall the direction of the light that passes through the eye. It passes exactly from the side of the vitreous body, and it comes from inside to the inner layer of the eyeball covering, and it comes to the retina. Here in this picture, vitreous body is here, and the upper pigmented layer is the most external layer. So light goes from this side, and you should remember about this, that light must pass first through ganglion cells layer, then to other layers, bipolar, and finally only then it comes to the photoreceptors. And even their photosensitive areas are directed not to the light but oppositely. And this picture is only schematic, and in reality the cones and rods photosensitive outer segments not simply directed to the pigmented layer, but they are submerged into them even. And this is very important because pigmented layer contains melanin, black pigment, and this black pigment absorbs the light, so there is no reflection back, and there is no chaotic distraction of light within the retina and the eyeballs and as a whole. So each light photons are absorbed by the pigmented layer. This is at least one function of the pigmented cells layer. But let's consider the other functions of these pigmented cells. Number of functions. First, due to black pigment melanin, there is a light absorption, so there is no light reflection within the eyeball, and it provides clear picture and clear difference between dark points in the visual field and bright points. Then it was found that pigmented epithelium is able to phagocytize the old discs of the outer segments of photoreceptors. This area, outer segments, now shown upward, they are almost inserted into pigmented epithelium, and really they consist of many discs. And specific experiments shown that labeled substances which are included into the disc gradually become included into the composition of the pigmented epithelium cells. And so it shows that all discs shedding constantly from the outer segments of photoreceptors are phagocytized by the pigmented epithelium. And then pigmented epithelium participates in the photopigments regeneration. In all these discs, the pigmented specific pigments are contained. In the rods it's called rhodopsin, and in the cones there are 3 different color pigments. But in all the regeneration process, constant recycling, the pigmented epithelium takes part. So you see that many functions are performed by this layer, which is not involved immediately into perception of light. Let's observe the difference between the fovea, here again we mean perhaps the central fovea mostly, and the periphery. Here this is the retinal periphery that contains, the more we come closer to the periphery, the more rods are located, and the very periphery of a retina contains only rods. And the closer we come to the fovea, the more and more cones appear between rods, and the fovea itself contains only rods. So here you can observe the convergence. You see all these, at least shown 5 photoreceptors, 5 rods. They come finally to one and the same ganglion cell. Convergence. Few of rods contact one and the same bipolar cell, and 2 bipolar cells stimulate one and the same ganglion cell. So this convergence does not provide high visual acuity, because you see here 2 light rays, and they are separated. These are 2 points actually of light, but we will not be able to differentiate this because one and the same ganglion cell is excited, and there is no distance between, so it will be appearing as one single point of light. And to the right, fovea of retina is shown. Cones are inserted here into pigmented epithelium, and these cones each contacts one individual bipolar cell. There is no convergence. Each cone contacts individual bipolar cell, and each bipolar cell contacts individual ganglion cells. Also, what can be mentioned, the cones here have some different shape from the cones out of fovea. Cones here are much thinner, maybe even thinner than rods, and it enables to pack tightly these cells in the fovea. So tremendous number of cones located in a very little space of fovea. And also it should be mentioned that here the thickness of retina is much less because the bipolar and ganglion cell layers are shifted to all sides from the fovea. So fovea provides coming of light almost directly to the cones, something like this. Then histology, macula luta and fovea histology. You can see in the middle the very fovea area, exactly because there is a pit, and you may see it resulting due to moving aside of the other cells other than photoreceptors. You see the light may come directly to the fovea. Thickness is much less here of the retina, and light comes in the area of fovea almost directly to the cells. You may see outer segments of the cells are located in this area, so much more light can enter this area. Visual acuity. We already mentioned that not all the retina has the same visual acuity, and this red peak reaches the highest visual acuity, labeled as 1 unit. It corresponds exactly to the highest visual acuity in the fovea by cones. This is produced by only cones, and it's called the so-called photopic vision, daylight vision. As in photos is light. Then rods, their acuity is shown by blue line, and you may see that average acuity does not exceed mostly 0.1, just 10 times less visual acuity. And also you can see, this is called scotopic vision. Rods are highly sensitive to light, and they can see in very low amount of light. So scotos is a Greek word for the darkness, and scotopic vision and vision in the night, but not full black, not 100% black, at least some light should be present. And you see here area of no rods, that's why rods have 0 acuity here, because rods are absent. And also you can notice the blind spot, the area where the optic nerve leaves the eyeball. This area does not have receptors. It's a very little area, and usually we do not notice even the presence of that one black spot in our visual field, because we constantly move our eyes, and so this little dot is not seen. But if eyes are fixed steadily, we can find this little area sometimes. This is a blind spot. It's normal. Comparison of rods and cones. Let's observe their differences. First, rods are highly sensitive to low intensity light. Partially it's because of their natural high sensitivity, much greater than for cones. And partially it's because many rods contact one bipolar cell, and finally few bipolar cells contact, especially in the periphery of retina, contact one ganglion cell. So finally ganglion cell is stimulated by many photoreceptors, and it increases the sensitivity of the periphery of retina to the light. Minimal light can be noticed by rods. So they are responsible for night vision. They have much lower visual acuity as compared to cones. They are absent in fovea. They adapt later. They adapt slowly, but their adaptation ability is greater than for cones, because of they can reach high ability to see the light when we have enough time of adaptation to the darkness. And finally, we can generally say that they're responsible for black and white vision. Then, as for cones, they have response only to the high intensity light. When light intensity seriously decreases, the cones cannot differentiate colors and cannot see well. So they're responsible for daylight vision. They have much higher visual acuity. They are present in fovea in 100% or almost 100%, and around fovea their concentration is greater than of rhodes. They adapt first. The adaptation does not reach very high level, but it comes faster than Rhodes adaptation. And they are responsible for the color vision, as we have 3 types of cones with different sensitivity to different wavelengths of light, so they are sensitive to different colors. And all 3 provide different degree of activation, provide sensation of each individual colors within the rainbow. Structure of the rods and cones. They all consist of course, the general portion of the cell with nucleus. They have the lower portion that faces to bipolar cell synaptic body. They have in the inner segment with some structures necessary for performing their function, which contains numerous mitochondria. And number 1, they contain outer segment, which consists of numerous membranous discs lined with rhodopsin or color pigment for cones. For rods, it's rhodopsin. And as you remember, outer segments are facing the pigmented cells layer, so that light that passes through outer segment immediately is then absorbed by melanin dark pigment. And these outer segments appear as numerous membranes disc, and the color pigments or rhodopsin pigment, they all are included into the membranes of these discs. And for rods, these discs are cylindrical, and for cones, that's why they are called cones, it's a conically shaped. Membranous disc with rhodopsin or color pigments. Then let's observe what happens under light to rhodopsin or color pigments. First we'll begin with rhodopsin. And you see, rhodopsin is the complex structure which consists of, it's located in the disc membranes, it consists of the protein opsin, shown in blue here, and specific portion, light sensitive portion, is retinal. Retinal without light under darkness has the shape of cis conformation, which makes this molecule angular. And so this is structure of the rhodopsin without light. But when light appears, it immediately produces isomerization, change of conformation from cis to trans. Let's observe once more. And you see, molecule of retinal becomes converted to the straight molecule, all trans retinal. This is universal reaction for any type of pigment, not only for rhodopsin, but for color pigments as well. The difference is only in the structure of the protein opsin, and the retinal is identical in all types of pigments. But due to difference in structure of the opsin, they are sensitive to different wavelengths of the light, and then they can respond to different colors for cones. So the basic reaction, conversion of cis to trans retinal, isomerization. This is what light provides. And after isomerization, which occurs very quickly, extremely quickly, a series of intermediate substances is formed. And finally, it forms the meta-rhodopsin 2. Meta-rhodopsin 2 is important substance that produces next conversion, and they go as a cascade reaction. Let's observe these reactions. Visual cycle in rods. You see, rhodopsin under light becomes converted to number of substances. I even don't think that it's necessary to memorize their names, and just what is better to remember, this is meta-rhodopsin 2, which is formed. And exactly this substance, meta-rhodopsin 2, produces next stage of reaction. It activates specific G protein, which is called transducin, which in turn activates next enzyme, phosphodiesterase of the cyclic GMP. And then it makes changes in the cell, which we'll observe in the next slides. But here you see that full cycle shows that there is recycling. Rhodopsin then should be formed again. It takes time. You see here written minutes. It takes time. And also you may notice here one more thing, that all-trans retinol also participates in the recycling of rhodopsin, regeneration, and this is known for us vitamin A. So you see that vitamin A is necessary for normal visual acuity of roads, and in case of severe deficiency of this vitamin A, people may develop something like something called night blindness. At night, in the dusk, the visual acuity falls to very low level because rhodopsin cannot regenerate normally in the roads, and for night vision exactly roads are responsible, and they cannot function normally in the case of deficiency of vitamin A. So let's observe dark current in the roads. Here you can see the greatest difference of photoreceptors from practically all other receptors. Usually all other receptors, in the absence of stimulus, have resting potential, and presence of stimulus produces depolarization. Here the situation is opposite. The rods and cones are depolarized in the darkness where stimulus is absent, no light. And this picture you can see in the middle, the membranous disc. This is schematic presentation of just one of the membranous discs with inserted into this membranous disc rhodopsin. Of course, the number of rhodopsin molecules is great, but here for schematic purposes you can see only one rhodopsin. And in the dark, let's observe what happens in the dark. Rhodopsin contains retinol in cis conformation, angular, and it does not form any intermediate substances, and there is no meta-rhodopsin 2, which may activate, located somewhere in the membrane, transducin, G protein. G protein is not active, and when it becomes active, it may activate the phosphodiesterase of cyclic GMP, but it does not activate in the darkness. So there is no activity of phosphodiesterase, which is able to destroy cyclic GMP and convert it into the non-cyclic molecule, simply GMP. And therefore, in the cell, the cyclic GMP is present, and cyclic GMP exactly is the substance that opens the channels for sodium in the membrane of the cell. So cyclic GMP gated channels become open, stay open, because cyclic GMP is present in the cell and not destroyed. These channels are for sodium. Therefore, sodium easily enters through the open channels, and this is something that produces dark current and depolarization of the cell. So the cells stay depolarized in the darkness. And actually, due to this, they release the transmitter. Let's observe what happens in case of light action. This is hyperpolarization, opposite case. See now, in the light comes to the rhodopsin and produces conversion of retinol from cis conformation into the trans conformation. And then, if you remember, many intermediate substances are formed, and it becomes converted finally to meta-rhodopsin 2. When meta-rhodopsin 2 is formed, it activates this transducin, G protein. Activation of G protein, hope you remember, results in release detachment of alpha subunit, and it can activate something. In our case here, phosphodiesterase of the cyclic GMP is activated, and phosphodiesterase splits cyclic GMP and converts it into simple GMP, guanylate monophosphate. Then channels which are cyclic GMP gated, they lose their ligand and they become closed. So sodium cannot enter anymore to cell, and it stops dark current, and it causes hyperpolarization, opposite situation. the pump regularly operating in the cell pumps sodium out, but sodium cannot come to the cell, and cell becomes more and more negative inside. Membrane becomes more negative with hyperpolarization. For better remembering this, let's observe these records of the photoreceptor potentials. See here, hyperpolarization of receptors was recorded. Actually, it's taken from a turtle retina. And you see, there many light flashes were used of different intensity, and so here you see the records of many stimulations with increasing force. And you see minimal hyperpolarization is achieved in case of lower flash, and most intense flash response was due to higher light. So this is light flash short term, just very short stimulation by light. And with higher and higher force of light stimulation, you see from minus 40 millivolt, the potential degree increases up to minus 65 millivolt, hyperpolarization. Membrane becomes more negative inside. And you can observe this number of different receptor potentials show that this is usual law of force as a characteristic for receptor potential. Greater stimulation produces greater response. But note again, this response is unusual. Stimulus light produces not D, but hyperpolarization. So let's recall all the mechanism of light action on rods. Light stimulates rhodopsin. Isomerization of retinal occurs and rhodopsin becomes active. As a result, it produces activation of the transducing G protein. Then G protein activates cyclic GMP phosphodiesterase. Phosphodiesterase destroys cyclic GMP by conversion into GMP, and it causes closure of cyclic GMP-gated sodium ion channels. When channels are closed, it brings hyperpolarization of photoreceptors. And naturally, it decreases secretion of glutamate by photoreceptors. The photoreceptors transmitter is considered to be glutamate, and when there is a depolarization, transmitter is released. Under light, there is a decrease of glutamate secretion. It's also unusual, but we'll consider how finally it creates normal response. Light should result in generation of impulses sent through the optic nerve, and in the absence of light there should be not stimulation of impulses. Note that this complex of biochemical reaction, complex biochemical cascade, provides great signal amplification. For example, just one molecule of activated rhodopsin, activated by light, can activate as many as 800 transducing molecules. Then you can imagine how many cyclic GMP phosphodiesterase molecules are activated, and so it's a great response. So actually it's the basis for very high sensitivity of rods to light. Let's see now the full mechanism. Rods in the darkness contain cyclic GMP. These molecules keep sodium channels open, and so dark current exists and cells are depolarized. And depolarization results in a continuous release of the transmitter, but this transmitter is inhibitory. Then everything becomes more or less clear. That bipolar cells are inhibited by rods in the darkness. Now, opposite situation. In the light, cyclic GMP is converted to GMP, channels for sodium are closed, and dark current stops. So no dark current, hyperpolarization stops release of the inhibitory neurotransmitter, which produces excitation of bipolar cells. No inhibition. Then, finally, ganglion cells become inhibited or stimulated. Again, in the darkness, rods, bipolar cells, ganglion cells line. In the darkness, depolarization produces release of inhibitory neurotransmitter, to the bipolar cells. Bipolar cells are inhibited and do not stimulate ganglion cells. As a result, ganglion cells do not send impulses through optic nerve, as it should be in the darkness. Then light produces, in the same line of cells, the following changes. Light stops secretion of inhibitory neurotransmitter from photoreceptors. Then bipolar cells release their excitatory neurotransmitter and stimulate ganglion cells. As a result, impulses are sent through the optic nerve and finally they reach the visual cortex and sensation of vision appears. If this appears to you complex, I must say that this is very simply explanation. Let's follow to the next portion of very complex explanation. It refers to the receptive fields of ganglion cells. Look, at the bottom of the picture you can see one ganglion cell, and it receives stimulation from many bipolar cells and finally from many photoreceptors. And all photoreceptors that finally stimulate one and the same ganglion cells, this full area of the retina can be considered the receptive field of one ganglion cell. And within this receptive field, we can differentiate two areas, the center and the surround. And it's so complexly organized that centers and surround can behave oppositely, and we differentiate two types of the ganglion cells, with on-center and with off-center. Let's understand what is this. When the spot of light is turned on for the center of the on-center ganglion cell, these neurons produce a burst of action potentials. Positive, it's stimulation. And surround produces opposite effect of inhibition. Then, if this is an off-center neuron ganglion cell, then the same stimulus, light, applied to the center produces decrease of rate of impulses, while the surround produces stimulation. So this is arranged very complexly, and each point of the retina contains numerous receptive fields with these types of ganglion cells on center and off center. It's much more complicated to understand, but it's not something for memorizing, it's for understanding mainly. Look, why we have these on and off center cells, these two types. They provide information about the changes, grades of the luminance of the amount of light. Luminance, if it increases or decreases in some areas as compared to others, especially compared to background. This is the most important information for such receptive fields of ganglion cells. And every point, as I already said, on the retinal surface is analyzed by ganglion cells having their on centers and off center. And this provides ability to detect stimuli with higher or less brightness as compared to the background. So practically this arrangement of ganglion cells, receptive field with center and surround oppositely in responsive, produces most of information about the boundaries of the objects in the visual scene. So the silhouettes, the boundaries of each object that makes the difference of color and light between the background and through the whole visual scene. It helps us to see well the boundaries of the objects, which are underlined, highlighted. Here this is an explanation of the neural basis, and again I should repeat that memorizing is not the purpose of this slide. It's necessary at least understand how it goes. It explains the background for a difference of signaling. In previous slides it was explained that generally transmitter glutamate secreted from the center, from the photoreceptors, produces inhibitory effect on the bipolar cell, but not always. This is classical for the on center, and let's observe this. Here, to the left side, we see the effect of photoreceptor activation or inactivation on the on-center ganglion cell, this left side of the picture. And now, let's observe how it goes for the on-center ganglion cell. Let's begin from the dark. If dark spot comes exactly to the center of the on-center ganglion cell, as a result, the photoreceptor cone in the darkness becomes depolarized, as we considered. Depolarization produces in the darkness release of the transmitter, and if this is inhibitory transmitter, it produces inhibitory effect on bipolar cell. Bipolar cells becomes hyperpolarized, inhibited, and this cell does not stimulate the ganglion cell, and ganglion cell with on center does not respond to the darkness by any impulses. So, and off center behaves oppositely. Then let's consider when light comes to the center of the on-center ganglion cell receptive field. Then light produces hyperpolarization of photoreceptor, of the cone in this case. Hyperpolarization stops release of the transmitter, and without release of transmitter, bipolar cell becomes depolarized for the on-center only case. And this depolarization results in release of excitatory transmitter, and on-center ganglion cell responds by burst of impulses. So, all as described in the previous slides, which generally explain the functioning of the retinal pathways. But for the opposite off-center ganglion cell, everything becomes reversed, because the same glutamate transmitter acts on the other receptors which produce stimulation. And I will not go through the full explanation, but you can observe that everything becomes reversed. Everything becomes opposite. But by now we consider only center response. But there is also surround. As for surround, everything is opposite to the center due to involvement of the horizontal cells, inhibitory cells. You see, surround cells connect not directly to the surround photoreceptors are connected to the bipolar cells not directly, but through the horizontal cells. So, and it provides the opposite effect, because horizontal cells are inhibitory and they release GABA, inhibitory transmitter. So surround respond of the receptive field respond oppositely to the any action of stimuli oppositely to the center, because all the results are reversed by horizontal cells, and due to inhibition everything becomes opposite. So of course it looks very, very complicated, but exactly this arrangement of receptive fields in retina helps us to see what we are able normally to see. The objects with their boundaries very well distinguished among the background about film objects. Let's move to the basis of the color vision. We have the 3 cone types, and they respond to different wavelengths. And there are 3 basic colors we respond to. These are blue cone. They are really not blue in color, but they are responsible for perception of blue color, green, and red. These 3 basic colors with 3 basic wavelengths we respond to. But each intermediate color produces activation of 3 types of cones in a different percentage. For example, you can observe here percentage. For example, for orange color, we don't have specific cones for orange color, but orange is located between yellow and red, and it excites to a certain degree red colors, the red cones to some degree green cones and doesn't excite the blue cones at all. And so different percentage of activation of each individual type of among the 3 cone types is unique for each shade of color. Each shade of color, and it gives us sensation of this specific color because it's differently activates each type of cells. Here you see the cones in fovea that are shown in their perception colors. It's done by special technique. Of course, the cells themselves do not have these colors, but a special technique colors them, and here they are colored exactly in their perception colors. So you may notice that perhaps most widespread are red cones. Also, high amount of green cones can be seen, and note that blue cones are much less in their number. Color vision abnormalities. It is not very often, but it happens for people to have abnormal perception of colors. It's because of defect of certain genes and therefore absence or defect of the certain type of the cone. This produces color vision abnormalities. Most often widespread is the red-green blindness occurs because of absence of red or green cones missing. Actually, in both cases, the people are hardly able to discriminate red and green colors. But actually, it's a different type. There are 2 different types. Absence of red cones, which is called protanopia, and absence of green cones is called deuteranopia. There are differences present between their capabilities, but both do not discriminate normally red and green colors, so they generally considered to be red-green color blindness cases. Blue cones are very unusual to be missed, and blue weakness occurs not often. Here you can see comparison of normal objects seen by normal person with trichromatic perception, and this is what can be seen by person with missing red cones, protanope, no red cones, and deuteranope with no green cones. You see, in both cases, little differences present, of course, but they do not differentiate red and green, which initially is very well shown on the first picture. So, how to detect the color vision abnormalities? The most widespread types you will study during the classes Rabkin tables, and these are Ishihara tests that also are developed on the same principle. They are based. The object is created by color only. In this case, mostly they use the digits, numbers, which can be differentiated on the basis of their color only. For example, if you make a photo of such a picture in black and white, then you will not see a difference. They will be random, scattered circles with no information about numbers. Numbers can be visible only due to differentiation of colors. But with absence of certain type of cones, differentiation will go differently. For example, for this, normal person reads 74. But person unable to differentiate red and green will read 21. Because exactly absence of differentiation of red and green colors. This is general test for any type of red-green color blindness. But if we need to differentiate exactly, red cones are missed or green cones, so in these cases the other types of pictures can be used. These pictures are numerous and they're used for full estimation of color vision abilities. Here, for example, a red blind person will read only number 2, but green cone missing person will read only 4, and normal person should read both numbers 4 and 2, 42. This is test for specific red blindness or only green color blindness. And again, it's necessary to mention that for precise testing, for correct testing, we need not to use the screen colors because they depend on screen characteristics, and it's necessary for proper diagnosis. It's necessary to use the originally printed colors on paper, which are which can exactly have corresponding shades of colors for correct testing. But approximately it can be tested using the screen, but very approximately. Now let's switch to the visual system conducting pathways. Mostly it's well known from anatomy. From each eye, optic nerve carries information from retina. Then there is the cross-section, partial crossing of fibers from medial halves of retina that form the optic chiasm. And the lateral portions of retina still carry fibers on the same side. Then optic tracts are formed after crossing. Then information goes to lateral geniculate body, and also there is passing of pathways through the quadrigeminal plate, superior colliculi. Then through optic radiation, finally, the pathways reach the visual cortex, which represents the central part of analyzer. Visual cortex has corresponding numbers according to Brodmann's numbering. All the cortex areas are numbered according to Brodmann, and area for primary visual cortex is area number 17. This is the most posterior occipital cortex. And numbers 18 and 19 are adjacent areas, so 17 is the primary visual cortex, and number 18 and 19 are secondary visual cortex, which is involved into deeper analysis of the visual information. Let's observe this. But first, primary visual cortex. You know from anatomy that primary visual cortex is arranged around the calcarine fissure. And here you can see this calcarine fissure goes deep into the brain, and this represents exactly primary visual cortex. Calcarine fissure passes deeply, and here there is an exact topographic presentation of each portion of the retina. Note that greatest area is taken for macula, including fovea representation. And then it goes more distant and more progressively distant from macula. So here it's like a map of retina present in the primary visual cortex. Direct conduction, like the screen with pixels, and with greatest representation of macula information, with highest visual acuity. But secondary visual areas also are involved due to analysis. Here you can see the representation of other functions. Primary visual cortex is smaller, but great amount of visual cortex is included, and even it comes partially to the parietal cortex, because it's necessary to estimate by the secondary visual cortex the form, shape of objects, their motion, direction, their three-dimensional position, and so on. And even some temporal areas also may participate in this function to estimate the visual details and to estimate colors, their change in the patterns of color, and so on. This all is function of the secondary visual cortex and adjacent, even associative areas. Optic pathways. Let's observe them from the beginning to the very end. Left sides of retina all come to the left side of the hemispheres, because of cross-section done only for medial portions. But then, if we consider not parts of halves of retina, but parts or halves of the visual field, it's all opposite, because of lenses that invert information, and all that we see from the right side, in the right side of the visual field, goes to the left side of retina, and opposite, vice versa. So right sides of visual field are reflected or conducted to the left halves of both retina in our eyes. Correspondingly, right halves of retina send their fibers all to the right side of the brain, right hemispheres. But the information sent from right halves of retina comes from the left halves of visual field. Again, this is all because of a lens which can convert rays of light to the opposite side. Optic nerve carries information from both halves of the visual field, But when it comes to the tract carries information only from one side of the visual field. So it's easy to memorize that left halves of retina send impulses to the left and right halves to the right. But if we consider not halves of retina again, just halves of the visual field, due to lenses, you see, it's all reversed and goes to the opposite side. Now let's observe examples of the lesions of optic pathways. First example of lesion is lesion of the nerve. Now in this case, this is left optic nerve cut. And of course, full left eye becomes totally blind. Left eye blindness. Nothing to explain, it's easy. The blindness is called also anopia. It is directly translated from Greek anopia, absence of vision. Then here, as example, we have the damage of right optic tract. Tract carries information already from two eyes, from both eyes. And the right tract has the information from the right halves of retina. But if you ask which halves of visual field are lost in this patient, of course they are opposite. Left halves of visual field. So if in case of tract damage, optic tract, the same side or homonymous sides of retina cannot see, cannot conduct information to bring to the visual cortex, but it corresponds to loss of the opposite sides of the visual fields, left. In this case, if left tract is damaged, so then left halves of retina will not be able to send impulses to the cortex, but it corresponds to loss of the right halves of visual fields. It's called homonymous hemianopia. Hemi means half blindness, but homonymous of the same side, left or the same right side. Let's continue to observe different damages. Now this is a damage exactly in the crossing area of fibers, medial fibers. And if optic chiasm is damaged, then it will be loss of the medial halves of retina. And now we can see that medial halves will lose the information from the lateral halves of visual field. Now it's not homonymous because they are left to the left eye and right side to the right eye. So it's called bitemporal because these are not nasal but temporal parts of the visual field. So bitemporal hemianopia. And last example shows the damage of the visual cortex portion. And as a result, some areas that maybe send impulses even to this cortex, but cortex cannot perceive these areas of the visual field become lost. And it is a dark spot, and darkness, scotus, darkness, corresponds to the scotoma. This is presence of black areas in the visual fields. Let's observe something more about local adaptation of photoreceptors. Generally, we know that in the darkness, photoreceptors, especially rods, become gradually adapted and their sensitivity increases. Local adaptation can occur also when part of retina is under light and part in the darkness, and the cells in the darkness should adapt. It doesn't often occur because usually our eyes constantly move. We don't look into one point. But if we do this, we can observe the so-called after images. Also, you can first let's consider the black and white after images due to rods adaptation. If approximately half of a minute you will look, for example, into middle star point without any movement of head and eye, so that the same areas of retina constantly have light, and the same area which has the image of this black ring will be constantly not lighted. Then the cells which are constantly under without stimulation by black ring, black color does not reflect anything, so there is no stimulation. And if really you manage to not to move exactly these objects in the retina, then if we look to the right side, the same point, you will see the brighter white ring, brighter than the background, which is also white, because the cells which we did not have the stimulation gradually within, let's say, half of a minute, become more sensitive to light, and then when you look at the same more or less white area, then you can see that cells with higher sensitivity perceive this white color as brighter color. So you can see after image, which converts like a negative, converts black figure into the figure of whiter, brighter color than the background. This is black and white after image. Again, I remind that usually we don't observe this because we don't look for a long time into one point. Most we move, and so there is no way, no time enough, not enough time for adaptation, local adaptation of photoreceptors. Here you can observe the simultaneous contrast. Let's see. If you see at this gray ring, you may notice that right side looks a little bit darker than the left one, but this is just because background is different. On the white background, the gray color seems darker, and in a dark background, the gray ring seems lighter. This is explained by these on and off centers, which show the bigger contrast between the background and shape. Look, now when this ring moves and totally becomes on the black background, it becomes equally gray, light gray. If it moves and becomes totally on the background of white, it becomes a little bit darker gray, but no difference in the ring itself. And then again, if it becomes located in the middle, usually it appears as brighter in the left side and darker in the right side. Then cones adaptation. Due to cones local adaptation, we can observe the colored after images. And here it's much more complex because colors are different, and you may hardly predict what happens, but there is actually possibility to predict that colors are changed from one to another. If you constantly look at a red object, the cones responsible for red color, if constantly they are stimulated by red color, they become less sensitive, and as a result the other colors, if you look then at white color contains all the wavelengths, and so it stimulates equally all the cones, but the red cones will not be so much active as they are tied by constant stimulation, and the color of the object will turn into green with little blue shade perhaps. Let's observe if for a long time, at least half of a minute, to look steadily on this star in the middle without any movement of the object on the retina. It's necessary not to move eyes, not to move head. And if it's long enough, then you may look at the star in the white background, and you can observe reverse. These two colors exactly exchange for each other. The heart becomes red colored and the background becomes green colored. This is the color after image. And the explanation of this may be given by the following opponent colors. This scheme, of course, is not for memorizing at all, and even not necessary to, but if you look through you can understand the difference. We have 3 types of cones, but only 2 types of ganglion cells become responsive to the lights, and the same ganglion cell, when excited, produces the sensation of red color, and when it's inhibited, it produces the green color. So as a result, you see red and green makes the pair of opponent colors which exchange one for another, and correspondingly yellow and blue. So it explains the presence of the 2 groups of colors, red, green and yellow, blue. And we can observe this on the following picture after images. But also it depends on shades, and sometimes screens cannot provide perfect coloring. But if you produce the same procedure, look straight on the dot in the center, spot in the center for half a minute at least or longer, without movement of the object in the retina, then if you move to the right side and look, the colors should be changed in places. Blue color should be changed into yellow and vice versa, and red and green will also change their places. But it depends on color of the screen, and then sometimes it makes them not exactly the same colors. The best way it can be done with a paper where every color is printed in exactly precise shade. But this just shows the complementary opponent colors. Blind spot revealing. There are many pictures that show the blind spot presence. Here from the screen I don't recommend to observe this, because with the screen on the computer it's hard to find it. The best way when it's printed in some book and you can find it. The task is to close one eye and with the other eye to see exactly on the cross and find such a distance from the picture that produces loss of the face. And brain simply, because it comes directly to the area of the blind spot. And brain simply fills this empty space by the same picture as surrounding. It looks like this. So it's possible when you use the paper printed picture of this type, when something disappears. There are many versions of such pictures which help to reveal blind spot, but I don't recommend to do it from the screen. And now, as we finished general consideration of the whole visual system, it's just a few additional slides just for perhaps partially amusement and partially explanation of some characteristics of our visual perception. These are visual illusions. There are classical illusions that show that we don't perceive exactly as it is many objects. For example, this is one classical illusion of the different lengths of this line with arrows. Arrows directed oppositely, and it makes illusion that the upper one is longer and lower one is shorter, but it's just illusion. In reality, there is no difference in size. Here, this is a classical illusion, and so-called Ebbinghaus illusion, where the central circles are of the same size, but when the circle is surrounded by smaller object and the other one by larger objects, the first one looks larger. Also, there is additional help to this illusion by the warm color and the colder color, so-called. But actually, really, this object is of the same size of this circle to the right side. Then another types of illusion can be visible next, where the squares are placed irregularly. They create the illusion of non-parallel blocks. But in reality, look, these are exactly parallel. And to the left side, you can see another type of, even it's not an illusion, this is the mechanism of brain finishing the unfinished picture. At this picture, you can observe the three figures with 3/4 of circle, but they are located in such a way that they create suggestion that there is a square placed in the middle. In reality, there is no square, but the brain just finishes the unfinished picture. It's often used by brain understanding of some visual field objects. There is also one well-known moon illusion. The moon located just above the horizon appears much bigger than the moon located high in the sky. And actually, there are many explanations for this illusion, and every such explanation is not considered to be full explanation without any objections. And this illusion is well known even from ancient times. But one simple explanation, not a final one, can be based on the estimation of the moon size relatively to the sizes of objects at the horizon. On the horizon, which is based on experience. The objects that are located very distantly from us, and they look similarly to the other objects located distantly, we can estimate as really very, very large in their size as compared to the objects of the same size located closer to us. They may create the same images size on the retina, but if they are considered to be very distant, we understand, which is based on experience, that these objects are very, very big in reality. And when the moon is close to distant objects on the horizon, and we know that the moon is much, much more distant from us, we estimate it's larger than when moon is located very high in the sky, where there is no object, there are no objects close for comparison. And practically it resembles somehow this classical illusion with surrounded by smaller object and surrounded by larger object, or nothing to compare to. This is illusion of non-parallel lines. Initially, you may see all lines are exactly parallel, but if they are crossed in such a way, there appears the illusion of non-parallel position of these lines. They become convergent or divergent from each other. Partially, some illusions can be explained again by the effect of perspective. When we consider that something is more distant, we estimate the object as really larger. And for example, to the left side, these two pieces, two lines, are of the same size, but the upper one always looks longer because there is an illusion of the depth of the space with perspective laws, when the parallel lines become progressively convergent in somewhere in distance, and this creates the estimation of the distance, and distant object of the same size appears larger for us. Then to the right side you may observe also a picture where this object looks like much larger than similar size object placed closer to us. This is also based on the estimation of the perspective, this illusion. This is based on the brain experience. And similar cases here, this figure very much distant, much more distant than the anterior one, but the size is the same, just we estimate according to experience. And similar illusion to the right side can be visible. This person is of the same size in reality than this one, but our brain estimates it as great difference because the anterior one is considered to be much closer, and so correspondingly distant is much bigger. And one more non-parallel lines illusion, which is also based perhaps on the perspective, because such a crossing of initially parallel line, look, perfectly parallel two lines, but with addition of these divergent numbers, it becomes like more distant in the middle, like biconvex almost. And there's one more color after image. If you look long enough to the cross in the middle of the colored picture, the reddish becomes normal. When you look to the right on the cross to the right, it becomes of red colored and leaves become green. This is again color after image based on Cohn's local adaptation. And this is the end. Hope that you understand more or less most of the information. Thank you for attention.